We have talked about the natural function of the immune system and the role of the MHC in the immune response. In this lecture, we will discuss the topic histocompatibility, or how to prevent a strong immune reaction which leads to a graft rejection. Two crucial factors are HLA matching of donor and recipient, and prevention that the patient will be transplanted in the presence of donor-specific HLA antibodies. Let's first have a look at the HLA matching of donor and recipient. Foreign HLA antigens on the donor organ are the most important targets of the immune response leading to graft rejection. Therefore, it is not surprising that the best transplant results are obtained with HLA-matched grafts. This slide shows that an increased number of HLA mismatches will lead to a poorer graft survival. The chance to find an HLA-matched unrelated donor is low, due to the enormous polymorphism of the HLA system. International collaboration via organizations like Eurotransplant will facilitate the search for HLA-matched donor organs. However, we have to accept that many patients will be transplanted with an HLA mismatched organ. If a patient is transplanted with an HLA mismatched graft, long-term graft survival is certainly possible, provided that the patient does not have preformed antibodies to the HLA antigens of the donor. Such antibodies can be induced by previous blood transfusions, pregnancies, or failed transplants. Already in the early days of transplantation, it became clear that transplantation in the presence of donor-specific HLA antibodies is associated with a high incidence of hyperacute rejection. In order to prevent this, a serological crossmatch is performed. The assay used for such a serological crossmatch is very similar to the one used for HLA typing. The next animation will explain the procedure.
Lymphocytes of the potential donor are incubated together with the serum of the patient in the same tray as is used for HLA typing. If the serum of the patient reacts with the HLA antigens on the lymphocytes, they will bind, and addition of complement will lead to lysis of the cells, which makes them permeable for the red dye. Such a positive crossmatch is considered a contraindication for transplantation. If the serum of the patient does not contain antibodies to the donor HLA, the antibodies will not bind, and addition of complement does not affect the viability of the cells. The red dye cannot penetrate the lymphocytes, indicative for a negative crossmatch, showing that this donor is suitable.
Rather than just waiting for the outcome of a crossmatch, the general policy is now to determine beforehand whether a patient has HLA antibodies, and if so, the specificity of the antibodies is determined. As soon as the HLA type of the donor is known, one can already exclude those patients that have proven antibodies to the donor HLA. For many years, this antibody screening was performed with the just described complement-dependent cytotoxicity assay. The serum of the patient is screened against a panel of HLA-type blood donors, and based on the positive and negative reactions, one can determine the antibody specificities.
The procedure is as follows. Every well of a Terasaki tray contains lymphocytes of different blood donors. When the serum of the patient is added, possible antibodies will only react with those lymphocytes expressing the HLA antigens recognized by the antibodies. This is not the case with the lymphocytes of donor A, and addition of complement will not lead to cell lysis and staining of the lymphocytes of this donor. The situation is different for donor B. Here, antibodies do recognize the HLA antigens of this donor, and addition of complement does lead to lysis and staining of the cells. By analyzing the results of such testing against a cell panel of about 50 different donors, one can determine the specificities of the HLA antibodies present in the serum. However, there is a problem associated with the use of this lymphocyte cytotoxicity assay, which is the fact that we are transplanting kidneys and not lymphocytes. Although the assay is very useful for the detection of antibodies to the HLA antigens, which are expressed both on lymphocytes and kidneys, lymphocytes also express other antigens which are not present on the kidney. If these antigens are recognized by antibodies in the serum of the patient, this will lead to false positive cross-matches and antibody screening results. In order to prevent this, new antibody detection assays have been developed. The serum of the patient is tested against polystyrene beads with only HLA molecules on their surface. If antibodies bind to these beads, one knows for sure that this antibody reactivity is directed against the HLA molecules and not against other irrelevant targets. The most specific antibody screening is the one using beads expressing only one HLA allele. When an antibody binds to a bead, one knows immediately the specificity, as beads expressing different HLA alleles have different colors. One can perform semi-quantitative antibody analysis by immunofluorescence. The more antibodies are bound to a bead, the higher fluorescence intensity. In this way, one can not only discriminate negative and positive reactions, but also the strength of the positive reaction.
We have seen that foreign HLA antigens are the most important targets of the immune response leading to graft rejection, and that the best transplant results are obtained with HLA-matched grafts. Thirdly, we have seen that transplantation in the presence of donor-specific HLA antibodies is associated with a high incidence of hyperacute rejection. In addition, now you are familiar with the principles of cross-matching and antibody screening. In the next lecture, important aspects for patient selection will be discussed.